Okay, it's 10 o'clock. We'll get started. Hello, and thank you to everyone around the world for joining us for this week's lab meeting. My name is Kristen Abood. I am the science editor at the Human Vaccines Project, and I will be your moderator today. This month marked yet another grim milestone in the COVID-19 pandemic as worldwide deaths due to the disease surpassed 5 million. In the face of this, many countries are still struggling to obtain enough vaccine supply. Only 5% of Africans have been fully vaccinated against COVID. Yet some countries, including the US, are struggling not with supply, but with vaccine hesitancy. The US now stands last among the 7 wealthiest democracies in vaccination rates, even as the FDA has approved boosters for certain populations who have received the Pfizer-BioNTech, Moderna, and Johnson & Johnson vaccines. According to the Moscow Times, Russia recorded its highest daily death toll from COVID-19 this week, which is attributed in large part to vaccine distrust in the country. But it isn't all bad news. Earlier this month, Merck and Ridgeback Biotherapeutics announced that they will seek emergency use authorization for their antiviral pill that was shown to cut the risk of hospitalization or death by half in those infected with SARS-CoV-2. If authorized, this would be the first oral COVID-19 therapeutic. Other good news came from AstraZeneca. The company is seeking emergency use authorization for prophylactic use of its combination monoclonal antibody product that was shown to reduce the risk of developing symptomatic COVID-19 by 77% in a phase 3 trial. If authorized, this would be the first antibody product available for COVID prevention. Before we begin today's presentation, just one note. The information presented today includes some pre-published data that is currently under peer review. At the request of our speaker, we ask that you not reproduce or disseminate the data presented. This session will be recorded and made available on our website and our social media channels for you to review. With that, I am very happy to introduce today's speaker, Dr. Peter Gilbert, a professor of biostatistics in the Vaccine and Infectious Disease Division of the Fred Hutchinson Cancer Research Center and the University of Washington. In this role, Dr. Gilbert focuses on the statistical design and analysis of clinical trials of candidate vaccines for HIV, SARS-CoV-2, and other infectious pathogens. He specializes in statistical methods and data analyses of these trials to understand how immune responses to vaccination and genetic features of infectious pathogens impact vaccine efficacy, so-called Immune Correlates of Protection analyses and sieve analyses. Dr. Gilbert is the principal investigator of the Statistical Data Management Center for the National Institute of Allergy and Infectious Diseases HIV Vaccine Trials Network. And he plays a similar role for US government-supported COVID-19 preventive efficacy trials, which is what we will hear about today. During the presentation, please send me your questions using the Q&A function in Zoom rather than in the chat. I will ask our speaker a broad selection of your questions after the presentation, and we will have about 25 minutes for discussion. With that, it is my pleasure to now introduce Dr. Peter Gilbert.
Yes, thank you, Kristen. I appreciate the opportunity to present these results to this group, and I'll share my slides. Okay. One more click. Okay. So I'll be presenting the immune correlates analysis of the Moderna vaccine for their phase 3 trial. So it's really a privilege to present these data because it represents the efforts of a large group of people. My work has been part of the U.S. government response team effort going back to about March of 2020, where our goal was to harmonize the design and the statistical analysis of the 5 different phase 3 COVID vaccine efficacy trials that were conducted through this public-private partnership approach. And Dean Folman has especially been my partner in leading the statistics for creating the harmonization and planning the immune correlates of protection analyses and implementing the analyses. And then there's laboratory leadership and clinical leadership as well. So in my role on the statistical side, it's quite important for us to do the research in an open, collaborative way that is reproducible. So we've been posting our statistical analysis plan for correlates for a long time now, and we have been developing all of the R computer code for implementing the statistical analyses in GitHub, so that it's visible. It's a public repository of code, and it's proven useful as a way to share how we do these analyses with people doing trials that aren't part of the USG effort. And this slide is listing these 5 different phase 3 trials that are through this public-private partnership. I think everyone knows what these trials are. And we're also hoping to do harmonized immune correlates analysis of other phase 3 trials that aren't part of the USG portfolio, so that we can maximize the amount of data and learning about immune correlates through a standardized analysis approach. And why are we doing this? Well, it's because we need to have a means for appropriately approving vaccines and for doing additional research on vaccines. And we know that the gold standard for evidence for approving a vaccine is a randomized, double-blinded phase 3, placebo-controlled trial. And thankfully that has been possible, and the way that primarily evidence has been generated to approve vaccines. But that's not going to continue. We know that with vaccines locally available, and that's increasing, placebo-controlled trials won't be feasible. And so an immune correlative protection is perhaps the most promising way to approve vaccines short of having a placebo-controlled phase 3 trial. And for example, the FDA has mechanisms of approval based on an immune marker surrogate endpoint. Basically, if there's evidence that the marker is established to reliably predict vaccine efficacy against a clinical endpoint of interest, then you can base traditional approval of a vaccine using the immune correlate. And when I say immune correlate, I really mean the same thing as a validated surrogate endpoint. And the FDA has a second pathway that if you have evidence that the marker is reasonably likely to predict vaccine efficacy, but not yet completely compelling, then it might be appropriate to make accelerated approval decisions and then confirm direct clinical benefit in post-licensure effectiveness studies. And so we can think of 3 categories of applications. One would be for an already authorized or approved vaccine, one could use the immune correlate for extending the indication to new populations. So this is a very contemporary application right now in the US, trying to authorize the vaccines for young children. Also, one could take a new vaccine candidate that's not previously authorized or approval and go for initial approval, either for refining an approved vaccine, such as adding a dose or putting in a new strain into the vaccine insert, or even trying to get de novo approval of a new vaccine where there's not even a version of that vaccine has been previously approved. So different levels of bridges to try to do based on an immune correlate. So this is the authorship of our preprint on this Moderna immune correlates analysis. And I'd like to thank the trial is called the COVE trial, so I'd like to thank the study participants and the Moderna COVE study team, and then to thank our Coronavirus Prevention Network colleagues, the Biostatisticians and Immunology labs. Thanks our leadership at the NIH and BARDA and the funding for this research. So the context of an immune correlates analysis is what was the overall efficacy of the vaccine. And we all know that the Moderna vaccine had about 94% efficacy overall against the primary endpoint of symptomatic COVID that's been virologically confirmed. And for approaching immune correlates, we divide the scientific investigation into 2 kinds. which we call these two kinds correlates of risk evaluation and correlates of protection evaluation. The correlates of risk evaluation, this is looking at the vaccine recipients. It's measuring their antibodies post-vaccination. And then it's seeing how well we can predict whether those vaccinees go on to get the COVID endpoint. And if we see a correlation or a prediction, then we say we have a correlative risk. Whereas a correlative protection analysis is looking at vaccine efficacy, and we're asking how well do post-vaccination antibody markers predict or cause vaccine efficacy against COVID. And a few more comments on why we do this division. So there's basically pros and cons of cores and COPs, such that we think that we need to evaluate both kinds, and kind of understanding the differences and the limitations of each can help us get a more complete scientific approach of understanding correlates. So the pro of the correlates of risk is that we can definitively get answers that are reasonably well powered, because it's basically standard statistical regression in a way. We're trying to, do association analysis. Do vaccinees with higher antibody levels have a lower rate of COVID? We can do this. But the problem is that if we find a strong correlation, it doesn't prove that you could use the marker as a reliable basis for predicting vaccine efficacy. And the reason is because we don't have exogenous control of the antibody level. We're not assigning the antibody level of individuals. Basically, we have an observational type study, and we could have confounding. It could be that vaccinees that have high antibodies also have really low exposure to COVID for some surprising reason. And if that was true, that would mean that an identified correlative risk may be misleading for decisions about use of a vaccine. So then the pro of a COP is that because we're trying to do causal inference and estimating how vaccine efficacy is linked to the immune marker in a causal way, we know that if we identify a COP, then we've got exactly what we need, that the answer is in terms of reasonably likely to predict vaccine efficacy. And then the con, or the challenge, is that with these causal methods, we cannot fully empirically verify all the assumptions we need for the inferences. And again, this is because we don't have exogenous assignment of the antibody level. We basically have to make no unmeasured confounding assumptions, or adjust for confounding, or do sensitivity analysis. So I think this means we're going to need cumulative evidence from multiple phase 3 trials, and external evidence from types of studies that have more experimental control, like challenge studies or in animals or humans. So for the COVE trial, the vaccine was given or placebo were given in 2 doses at day 1 and day 29. We're studying antibody markers measured at day 29 and day 57 as correlates. And basically we have data on follow-up through 4 months post dose 2. And it's all during the blinded period of follow-up. So we have the full rigor of the randomized placebo-controlled trial. And so we're only studying the early antibody markers as correlates of COVID through 4 months. Later on, we'll be able to study correlates for longer term endpoints. We used a 2-phase case cohort sampling design for measuring antibody markers. And what's done here is, first we take a stratified random sample of the entire study population with stratification by age, at risk factor, and underrepresented minority status, and also by baseline SARS-2 negative or positive. And we measure the antibody markers in that stratified random sample. And then what's done is we also measure the antibody markers in all of the vaccine breakthrough cases, because they're the group that has the richest information to learn about correlates. And we do the correlates analysis in the same population for which the primary analysis of vaccine efficacy was done, which is the per protocol baseline negative individuals. These are individuals who received both doses of vaccine without major protocol violations and didn't have any evidence of previous SARS-2 infection. Now, so our lab colleagues, Adrian McDermott at the VRC and David Montefiore at Duke, they work tirelessly to build and validate assays for measuring binding antibodies to spike or to the receptor binding domain of spike, or to measure neutralizing antibody, ID50 or ID80 titers to the D614G strain of the virus. And importantly, both assays the readouts were put onto WHO international units, which is going to be important for being able to compare the results of the study to the results from other studies. So we approach the statistical analysis with multiple different frameworks of statistics. The correlative risk framework is probably what people are most familiar with. I mentioned it's regression analysis, association analysis. So we do things like Cox modeling regression with adjustment for covariates, machine learning to see how sets of markers best predict COVID. And then the bottom part of this slide is showing the correlative protection analysis. There's effect modification analyses where we're asking how does VE vary across subgroups defined by their immune marker level in vaccinees. And then we have interventional mediation causal methods. There's the classic causal effects framework where we imagine that everyone was assigned to have the antibody level to a certain value. And we estimate risk of COVID under that assignment versus under assignment to placebo. And that's giving us a picture of how vaccine efficacy depends on the antibody level. And then there's stochastic interventional vaccine efficacy, which I won't show today for the sake of time. And then I will show results in mediation. But this is basically pathway analysis. It's asking how much of the vaccine efficacy against COVID is through a given immune marker measurement at day 29 or day 57. And so this is really just a graphical depiction of the different types of statistical frameworks we use for correlates analyses. And the green circle ones are the frameworks I'll be showing results for today, the darkened ones. The light green ones are the ones where we're finishing up our answers now, and we're going to be reporting those a bit later. And this is to say, later on, we'll do what we call stage 2 correlates, where we'll look at the antibodies over time, over the 1 or 2 year follow-up period, and handling things like the fact that placebo recipients crossed over to the vaccine arm, and now there's a dose 3, and do outcome proximal correlates, where we're looking to see how does the current level of antibody correlate with the instantaneous rate of the COVID event. And I won't be showing results on these today. We don't have enough data yet. We need more antibody assays need to be run to get these data. But so this is just forecasting for the future. So what is our data set for correlates? So you won't be able to read this, but basically this is the vaccine arm of the study. And the point I want to make is, this is showing the result of our 2-phase case cohort sampling for antibody measurements. And basically the box at the bottom is showing the different strata where we got antibody marker data defined by age, at risk status, and minority versus non-minority status. And then the blow up box on the right is showing the total amount of data we got. So we got data in the random subcohort for about 1,000 individuals, day 1, day 29, day 57. So it's really quite a large data set of imagicity. And then the most critical data were the vaccine breakthrough cases, where we had 46 breakthrough cases with data 7 days after the day 29 marker visit, and then 36 vaccine breakthrough cases starting 7 days post day 57 visit. And we start 7 days post to try to make sure that our antibody measurements were made by, the antibodies that present were made by vaccination and not made by a natural infection that happened, very near the date on which the antibodies were measured. So this slide is showing the ID50 titer on the y-axis, and each vertical bar here is one vaccine breakthrough case. And the only point I want to make on this slide is that the cases occurred approximately uniformly over the 4-month follow-up period post-dose 2. And this slide is depicting the variants that were present in the the region. The study was done in the United States during the follow-up of the trial. And so the right table is Hamming distances, and then the left graph is from next strain. And the point I want to make here is that the viruses that were circulating and causing infections in this trial were really quite close to the vaccine insert. About 70% of the viruses were within 1 or 2 amino acid mismatches from the vaccine insert. And so this whole study was pre-Delta. We're essentially getting a clean look at what was vaccine efficacy and what are immune correlates against Wuhan-like viruses, with a small exception. There's maybe 10% of the infections are with viruses that are more distant from the vaccine insert. Some demographics, and really the point I want to make is that 54% of the participants for whom we measured antibody data were from communities of color in the United States. It was intentional to oversample communities of color to ensure enough characterization of imagicity in those groups, so we can get direct data on predicting vaccine efficacy in those subgroups. This slide shows the correlations of the 4 antibody markers that were studied. This is, I'm going to mostly be focusing on the antibody markers at the day 57, 4-week post-dose 2 time point. And we're studying 4 markers here, as I mentioned, the IgG to spike or to RBD, and then the neutralization ID50 or ID80. And the first point here is that the 2 binding antibody readouts are very highly correlated. And similarly, the 2 neutralization measurements are very highly correlated. So this means that we can essentially only look at binding antibody to spike and neutralization ID50 and get all the information. So I'm only going to focus on reporting those 2 markers, 1 binding, one neutralization. And another point is that the binding and the neutralization markers are reasonably correlated, with a correlation of about 0.73 to 0.80. Okay, and this is to say that, I mentioned that, the Achilles heel of correlative risk analysis is that you could have confounding. And so therefore, all of our analyses adjust for baseline factors that are predictive of COVID to the best of our ability. And so what we did is we first used machine learning to build a risk score for COVID. And we built that risk score in the placebo arm, where we had the, luxury about 600 endpoints. And then we used that model built from the placebo arm to define a risk score for every vaccine recipient that is included in the correlative risk analyses. And also we adjust for the same variable in correlative protection analyses. So let's move on to results. So kind of one of the most straightforward ways to think about correlates is we want to compare the antibody markers in the vaccine breakthrough cases versus the non-cases who never acquired the study endpoint. And this table is showing data for all 8 markers as defined by the 4 markers and the 2 time points, day 29 and day 57. And the result is that, consistently across those 8 markers, the geometric mean concentrations or titers are always lower in the cases than the non-cases, with the confidence intervals for those geometric mean ratios being below 1. This slide shows the raw individual data points for the binding and the neutralization readouts left and right. And I think for time, let's focus on the day 57 panels, the right panels. So if we look at both the spike and the neutralization, the purple violin plots are above the orange violin plots. So that's showing that the antibody readouts are higher in the non-cases than the vaccine breakthrough cases. And you see a lot of spread in these data, and a lot of overlap too. So we're not getting a very clear so-called threshold correlate where most of the non-case data points are above the case data points. But you'll see we are seeing correlates in that definitely there's higher risk for those with lower antibodies and higher vaccine efficacy for those with higher antibodies. Yes, and getting into those statistical inferences. For a Cox model, adjusting for covariates, what's shown here are hazard ratios for each 10-fold increase in the antibody marker, either measured at day 29 or day 57. And basically these hazard ratios are all less than 1, with the upper 95% confidence limit well below 1, which is a way to say that indeed higher antibody is associated with a lower rate of COVID consistently across all the markers. And we can look at the results of this Cox model in terms of absolute risk on the y-axis. And so it's showing, again, in another way that vaccinees with higher antibodies have a lower rate of COVID-19. And we're going to start to see an issue here that we need to pay attention to, that on the left of these curves. So the green histograms are the distributions of the antibody markers in the vaccinees. And you can see that there's not many vaccinees with low titers. And so that means there's less precision of the estimates of risk in the left tail. So other correlates analysis we did is we divided vaccinees into low, medium, or high antibody groups, but by tertiles. And we found that indeed the vaccinees with the upper tertile antibody levels had a lower rate of COVID than vaccinees with lower antibody tertiles. It tended to be about a 4-fold difference in risk. That was consistent across the markers. And this is showing what I call nonparametric threshold analysis, where what's now being shown is for any given point on the x-axis, we're defining a subgroup as vaccinees that have antibody level above that threshold. And then the y-axis is reporting, the estimated, cumulative incidence of COVID for that threshold subgroup. And basically you can see that obviously, risk is decreasing as you make the subgroup smaller with a higher threshold. And we're not really seeing a cliff that there's a given, threshold above which risk is 0, except maybe for a very small percentage of the vaccinees. But to me, this analysis supports a continuum model that the higher the antibody level incrementally, the greater the vaccine protection. And this analysis is fully non-parametric, so it's really letting the data teach us without accidentally mathematically assuming a shape. So this is the correlative protection analysis looking at vaccine efficacy as a function of the antibody marker. And so you can see that there's a monotonicity here, that higher the antibody level, the greater the vaccine efficacy. And again, we have to take the left tails of these graphs with a grain of salt because there's not many vaccinees with antibody data. So there's weak precision in the left tail. So to me, the real point of this graph is that in the range where there's a reasonable amount of antibody data, like if you look at the neutralization on the right, from about titer of 50 to about 2,000, the vaccine efficacy increases in that range. And I think that's a pretty solid result from this analysis. And then the other type of the correlative protection analysis that we're reporting is the causal mediation analysis, where this is a type of causal inference that's been used in biomedicine for a long time. And the idea here is we basically imagine that we could deactivate the neutralization titer down to what it would have been had the person gotten placebo. And because everyone started, SARS-2 naive, what this means is basically totally removing the neutralization response, but not taking away any other consequences of vaccination. And then a perfect mediator would mean that after you delete neutralization, you have no vaccine efficacy left. So in this analysis, which we were able to do for the day 29 ID50 9088 titers, we see that after deleting neutralization, we still have 56% efficacy left. So that means the neutralization titer mediated 68% of the vaccine efficacy, but not 100% of the vaccine efficacy. So I think we're seeing evidence that neutralization is a partial mediator of vaccine efficacy, but not perfect. So let's discuss these results. So first, a summary of the correlative risk findings. I've mentioned that basically consistently across all the antibody markers at both measurement time points, we're documenting correlative risk, and we passed the pre-specified family-wise error rate multiplicity adjustment. So that's a solid result. And then for correlative protection, we're finding evidence of higher antibody titer corresponds to a higher vaccine efficacy. For example, at a titer of 5 in international 50 units on the WHO scale, the efficacy was 71%. And then at a titer of 1,000, it's up to 96%. So it's about a 5-fold increase in vaccine efficacy going from a titer of 5 to 1,000. And if I were to say one kind of practical take-home point of this correlate study, I would look at this plot here. And we can note that a titer of 100 international units per milliliter was associated with greater than 90% vaccine efficacy. And to put our mediation findings in context, where we found about 68% of the vaccine efficacy mediated, there's been mediation analyses done for the inactivated influenza vaccine. And this paper estimated that about 57% of the vaccine efficacy is mediated by the HAI titer. And interestingly, HAI titer has been used as a surrogate endpoint for approving flu vaccines for many, many years for updating the flu strain each year. So it shows that for COVID, we're already in the ballpark of the quality of a surrogate, partially valid surrogate endpoint that might be able to use for certain approval decisions. So, an interesting question is, do neutralizing antibodies fully mediate the protection? And, I've mentioned that, well, if the antibodies did fully mediate protection, we would see that there would be no vaccine efficacy for vaccine recipients with undetectable neutralization titer. And we didn't see that. We saw a point estimate of 50% efficacy for the subgroup with undetectable titer with a wide confidence interval. But I think actually we're not quite able to precisely assess mediation for the day 57 markers because we had only 2% of the vaccine recipients having undetectable new titers. So I think realistically, we're gonna have to wait for like AstraZeneca and the ensemble studies and other studies where there's a greater fraction of the vaccinees that never achieve detectable new titers to be able to get a more secure handle on this issue of mediation, at least fully empirically. So it's likely that additional markers are needed to mediate 100% of the efficacy. It's actually possible that neutralization as a biological function mediates a large proportion of the efficacy, but the particular biomarker that was used, an ID50 estimated from a neutralization curve, it might not have been maximally sensitive. We could have used a partial error into the neutralization curve, or there could be a way to configure the assay to be more sensitive to pick up the lower level neutralization titers that could be protective. And of course, there could be other immunological functions like Fc effector functions or Tcells that are important that we're not including in these analyses. Also, we only looked at serum markers. It might be the mucosa is important. And also, we're not accounting for anamnestic responses that aren't fully represented by a single time point measurement. Now, so to put this evidence for a correlative protection in context, this slide is summarizing other lines of evidence for neutralization as a correlative protection. So there's meta-analysis, which has shown that phase 3 trials where vaccines had higher efficacy also tended to have higher new titers. So that's pretty strong evidence. Also, there's been similar correlates findings in other studies, and I'll mention that on the next slide for a AstraZeneca UK study. And importantly, there's a non-human primate vaccine challenge study of the identical Moderna vaccine using the identical immunological markers that showed that basically the binding antibodies and the pseudoneut antibodies were a correlative protection in this challenge model, both for subgenomic RNA levels in the lung and in the nose. And also they passively transferred antibody, a purified IgG into hamsters and then challenged them and found that they got a correlative protection. So when the ability to have that kind of experimental challenge, provides some insight into mechanism of protection. And then there's been other lines of evidence from natural history studies and also the proof of principle from the broadly neutralizing monoclonal antibody studies. And this slide is really elaborating the challenge study of the mRNA vaccine that I just briefly mentioned, just showing the results about neutralizing antibodies and IgG being correlates of protection, both in the non-human primates themselves and also after passive transfer into the hamsters. So then my colleague Marian Bassi, she has led the correlates for the Oxford AstraZeneca vaccine, and she put together this interesting plot where basically our results for Moderna are the black curve of vaccine efficacy by new titer, and then the Oxford results are in green there. And we were able to do this comparison because we put the assay results on standardized units, which is I think a great thing to do. And you can just see that in the range where we have antibody data, we're getting about the same levels of vaccine efficacy. So this is encouraging that we might be able to get a cross-platform neutralization correlative protection. And this is a study in healthcare workers in Israel, real-world effectiveness studies, for the Pfizer vaccine, where they also found evidence for a correlative risk. So that's encouraging too, that there's consistent results with the Moderna results. And then this is kind of a statistical point, but as a statistician, we plan the study to look at both day 29 and day 57 antibody markers as correlates. This is a bit non-traditional because usually what people would do, they would just look at the day 57, which is the 4 weeks post second dose, and study that as a correlate. But we wanted to look at day 29 as well because it would be very practical to get a surrogate endpoint even earlier than day 57. And so we think about it empirically, can we predict vaccine efficacy just as well using the day 29 without having to do the day 57? And it turned out the data are supporting that, the day 29 data are indeed as good a predictor, maybe even a little better than the day 57 markers. So if this is confirmed through more studies, it could lead to a more practical surrogate marker. So some limitations, our Cox modeling analyses, they assume a parametric shape of how COVID-19 risk and how vaccine efficacy depends on the antibody marker. So it means we need to take the point estimates at specific titer values with a grain of salt. And we did also apply more flexible generalized additive models and got similar results. And also, we have fully non-parametric methods for estimating vaccine efficacy as a function of titer, which we've started to apply. And so we'll be able to report those results soon. And generally, we're seeing consistent results. And then, I've alluded to the fact that in these field trials, because we don't have experimental assignment of antibody levels, and also we don't have vision for what the exposing titers were. Basically, there's a lot of variability in challenge dose. It means that we cannot really prove mechanistic correlates from these field trials. We basically can study correlations and then study causation under assumptions like no unmeasured confounding. But if we really want, true mechanistic causal evidence, we have to go to like the challenge studies to support that. And thankfully there are such things that do bring in that support. And then so, as the SARS-2 vaccine field continues to, generate evidence on immune correlative protection, these are some of the questions that the field is, wrestling with is what to do with these neutralization markers. Basically, what bridges do we feel can be crossed based on these markers? And should we do it with the traditional approval or provisional approval? I think essentially this issue of going down in ages is indeed being handled with neutralization markers plus other data like safety data and dosing data. And then there's issues about putting in new variants into the vaccines and approving new vaccines where it wasn't possible to do a phase 3 trial, et cetera, where a lot of discussion is going to be needed. And then just to mention, to close here, a few next steps of research toward understanding correlative protection. So this study we've been able to do so far, it studied early binding and neutralizing antibody markers as correlates of one particular endpoint, which is symptomatic COVID, over about short-term 4-month follow-up for viruses that were similar to the vaccine strain. So this leaves many other things to study, such as COVID much longer after vaccination, or taking additional antibody measurements, including to the Delta variant, and integrating in features of the virus into the correlates analysis. And so this is the schema of the amended protocol from the Moderna study. So the PDV is the participant decision visit. That's when the placebo recipients and the vaccine recipients, they're all brought back and blood was drawn and an RNA test was done for SARS-2, and the placebo recipients were given vaccine. And now there's continued follow-up, and there's going to be a dose 3 that most volunteers, I think, will sign up for. And so we'll be able to get antibody data longitudinally over the study and measure seroconversion and measure RNA positivity. And then for new vaccine breakthrough cases that we see, we'll be able to fill in their antibody marker data later to do more correlates analyses. And indeed, to summarize, what we are planning for additional immune correlates is, one of the principles of correlative protection is, protection is specific to a phenomenon. So just because you have protection against severe COVID doesn't mean, or you have a good correlative for severe COVID doesn't mean you have a good correlate for asymptomatic infection or for viral load in the nose. So we're going to be looking separately at correlates for the secondary endpoints of these protocols. And I mentioned we'll include vaccine breakthrough cases that are occurring over the full 2 years follow-up. And then we're going to integrate the SARS-2 feature data into the correlates analyses. So we'll do like Delta specific correlates where we'll look at antibody to Delta as a correlate of COVID with Delta and infection with Delta. And we'll look at how correlates of risk depend on the distance of the SARS-2 virus to the vaccine insert, where distance could be measured either by the amino acid sequence, like a Hamming distance, or it could be measured by the measured sensitivity of the virus to vaccinee sera, or by a predicted sequence score of how sensitive it is expected to be to vaccinee sera. And also, we can do sieve analysis where we directly look to see how does vaccine efficacy depend on the features of the virus. And where we discover features that modify vaccine efficacy, we can look at correlates of risk, segregating by endpoints defined by different levels of those features to get additional insights into correlates of protection. And then, we'll see if we can get other antibody data, like efficacy effector function data, so we can do more machine learning to look at, can we improve the correlates by adding in additional functions? And then T cells, we know we can't do these. We didn't store PBMC in the studies. That would have been kind of infeasible given the scale and the speed. So we can still study T cells as correlates by meta-analysis when we get several phase 3 data sets together. Yes, and thanks again to my Coronavirus Prevention Network colleagues that worked on these analyses together, and to the many colleagues and sponsors. Okay, I'll stop there.
Great. Thank you so much, Dr. Gilbert. Really appreciate you taking us through all of that data. And we have quite a few questions coming in for you. So maybe we'll start with the question on the Q&A about whether you're considering T cell responses as markers of protection as well.
Yeah. So yeah, I think that is important. So I mentioned that none of these 5 phase 3 trials were kind of bold enough or foolhardy enough, I don't know which one to say, to try to collect PBMC in all the study participants, because there was such a premium on speed of these trials to get answers, and then setting them up and logistical simplicity and all this. So that means we can't do this type of case control correlates analysis. In other words, we just won't be able to measure the T cells in the vaccine breakthrough cases because we don't have the PBMC. The AstraZeneca trial is a bit of an exception where at least they have a subset of the phase 3 trial participants where they did store the PBMC, so at least be able to characterize the T cell immunogenicity in the study. But then the way we're going to plan to study T cells as correlates is through the meta-analysis approach. Maybe I'll go back to that slide real quick, because I think it is very important to try to include T cells. Yeah, where did I put the meta-analysis slide? Well, okay, the picture on the lower left there, it says COP meta-analysis correlative VE. This is the way in which we can study T cells as correlates, where what we'll be able to do is from phase 1 studies of vaccinees where PBMC were collected, by designing those studies to look at essentially the same study population as was used in the different phase 3 trials, we can characterize the group level T cell response. And then we'll have the vaccine efficacy estimates in the phase 3 trials. So we'll be able to directly study whether vaccines that have higher group level T cell responses also have higher vaccine efficacy. And we'll be able to also study antibodies integrated into that same analysis to try to tease out what's the interrelationship of antibodies and T cells to the level of vaccine efficacy. And then I think there's some surrogate ways that people are trying to get at T cell responses with, there's been some literature on assays that can be used on, the blood samples that we did store that are highly correlated with T-cell responses, we might be able to try those analyses.
Right, right. Okay, we have a couple of questions on the day 29 data, which you said was as if not stronger link to the correlates. Do you think that that would apply to every approved vaccine as well as after doses 1, 2, and 3? And do you think that day 29 data could be used as a long-term prediction of protection?
Yeah, I think it's an empirical question, and we only have this result from one study at the moment. I think if we see a consistent pattern that you can predict risk and efficacy as well using the 4-week post-dose 1 versus the 4-week post-dose 2, then that'll start to build a case that it's possible to do this. And I mentioned, because we'll be looking at vaccine breakthrough cases for the full 2 years, we will be able to directly compare the prediction ability of the day 29 versus the day 57 marker. And we should be able to do that across the 5 phase 3 trials, so we can at least judge whether it's appropriate to do that or not.
Right, right. So we have a question on the neutralization protocol you used and how that compares to the one used by the manufacturer of the vaccine.
Yeah, I might not have a great answer on that question, because I wasn't close enough to the assay development.
Okay.
I can say that David Montefiore tends to emphasize that assays by different labs tend to be quite different. That it's still a little bit like the Wild West with that's the term he uses, of getting the assays standardized or understanding how to compare the assays, and I think that's one of the reasons why using WHO international standards is so important. Like basically you can't read a paper that used a completely different lab and different assay and look at the readouts and compare them to to our results using international units, and unless you go that other paper, you can get that lab to go back and put its units on the international scale. So
Right.
yeah, this is a pretty serious issue, that if the scientific enterprise was kind of perfectly functional, then all these studies would be, well, I mean, it's kind of impossible because it takes time to validate assays and build these standards, but it'd be a beautiful thing if we could find a way to get all of these studies on the same scale. Yeah.
Right. So you spoke about the challenge models that could be used to enhance the correlates analysis. Do you consider the NHPs to also be a good in vivo model for prediction?
I mean, I think it's, I mean, again, I'm kind of, because I'm not, a person that runs these studies, I'm not, I can say I work very closely with people like Dan Baruch that have done a lot of work in the challenge models. And, I think they're very useful, extremely useful actually, because we haven't really been getting much data yet from the human challenge model. And we have been getting quite a bit of data from the non-human primate challenge model. And I think the fact that, like the mRNA vaccine, we're seeing correlates in both the non-human primates and in the human studies is some validation in the model. And actually, I think what we probably can do is take these US government assays and apply, do more of this thing that we've just done for Moderna and apply the exact same assays on the non-human primate samples, so we can start to draw the kind of the vaccine efficacy core protection curves in animals and quantitatively compare them to humans, and over time we can understand how we can quantitatively bridge the models from primates to the humans.
Right. Well, and given the focus on boosters, we have several questions asking you for your opinion on how the data that you've presented will suggest that maybe people will need additional boosters beyond the 3rd dose, or whether you think the heterologous prime boost regimens can improve the efficacy of vaccines.
Yeah.
It's asking a lot of you.
Yeah, no, yeah. So what we've been, I've mentioned what we've been able to study so far is, an early antibody marker as a correlate of disease over the next 4 months. And we know that these antibody markers are picking up, antibody concentration in the peripheral blood, and they're not measuring memory B cells or something like that. So, what's going to happen 18 months after vaccination or after dose 3? It's kind of getting into questions for which we don't have data yet. But we are going to be able to study these early markers as predictors of risk of COVID and infection over about 12 to 15 months, post vaccination. So we'll at least be able to understand much better, how these peripheral blood markers can accomplish that task. But I think if one could try, if we tried to come up with a translation of our main result to this question of when to boost, let's look at this. If we look at this rightmost plot here,
right,
we see that a titer of 100 corresponds to 90% efficacy. So one could say, well, okay, when your titer goes below 100 or goes below 50, when the efficacy starts to get below 80 or 70% or something, then you better boost. That's a bit of a leap, again, because it's a peripheral blood marker, and it's a different question of what's happening 6-7 months after vaccination based on your current antibody level at that time point.
right.
But I've heard people say that it might be a conservative strategy to do this because probably the memory B cells are going to be there to some extent, and what's in the periphery is kind of an underestimate of the true immunity that's available to the person.
Right. So we have one question asking if there are any real world examples where the correlate analyses were ultimately very inconsistent with the mechanism of protection.
Well, yeah, I think I don't know if it's been proven, but people often refer to the herpes zoster vaccines, mm-hmm. where there's papers that state that Tcells are the mechanistic correlative protection, but binding antibody concentration is a adequate non-mechanistic correlative protection that can be used to statistically predict vaccine efficacy. I'm not sure. I haven't ever seen any smoking gun evidence that that was absolutely true, but that's an example that's often cited. Okay.
So have you factored any impact of age into the models of the correlates, and is that possible? We have a question specifically, is a neutralizing antibody titer of 100 the same in a 30-year-old as it is in a 70-year-old with respect to efficacy?
Yeah, so we can't study whether the correlate differs by age yet, just because we don't have enough vaccine breakthrough cases. So everything we did so far is pooling over all the age groups. We did include age as a baseline covariate to adjust for confounding, because we know that age actually very well could be a confounder because it probably affects the immune response, and also we know it affects the rate of COVID as well. So yeah, but I think later when we can pool data across the studies, we can look for modification by age. And the other thing to say that in a way, the fact that the level of immune response varies across the ages is part of the way that we identify a correlate, because including a wide age range leads to a greater diversity in the immune responses, and that variability in the immune responses across vaccinees is the key thing that's needed, for correlating with outcome.
Right. We have a question asking if you think the correlate of protection for the vaccines could be different than it is for the correlate of protection in natural infection.
Yeah, that's always an important question. So I think when we, again, because we only could do correlates for the, SARS-2 naive individuals, we're basically getting a clean look of, correlates for vaccinees. To be able to answer that question directly, what we would want would be these phase 3 data sets over the full follow-up period where we also included the SARS-2 non-naives, the previously infecteds into the analysis. And if we had those, what we could do is we could study what is the relationship between antibody and risk in placebo recipients that were naturally infected, and compare that to the relationship between antibodies and risk in vaccinees, and basically, empirically study whether those relationships are the same based on natural infection versus vaccination. So I think we have, long-term plans to be able to do those analyses, but we just can't do them yet. I think dengue is an interesting example where there has been a lot of correlates analyses over the years that have dealt with this issue, that some of the vaccinees or placebo recipients are naive or non-naive to dengue.
Right.
And we've tried to tease out the differences between the naturally induced antibody and the vaccine-induced antibody as correlates.
Right. Well, thank you so much, Dr. Gilbert, for that excellent presentation on what is very important and interesting work. And we really appreciate you taking the time to share the presentation with our audience and for answering our questions today. I would also like to thank all the attendees for participating in today's webinar and for submitting such great questions for our speaker. We are always fortunate to have such an engaged audience for these lab meetings. And I invite you to join us 2 weeks from today on November 4th for our next lab meeting. Our speaker that day will be Dr. Julie Gerberding, Executive Vice President and Chief Patient Officer at Merck & Company. And her presentation will focus on pandemic preparedness, products, platforms, and partnerships. It's a lot of P's. And if you're interested in more research on COVID-19, please sign up for the HVP COVID Report, a bi-weekly newsletter that provides insights from experts around the globe and highlights the latest scientific articles and data. And finally, please visit our website and follow us on LinkedIn, where we will upload a recording of today's webinar, which I know there were a lot of questions in the chat about where you can get a copy of today's presentation. So please check our website and also on LinkedIn. And with that, I'll say thank you again for participating today. Stay safe, and we hope that you'll join us again on the next Global COVID Lab meeting. Thank you so much.